So we left off our story of making protein after the process of transcription. So transcription went from the DNA to the RNA. And then we talked about how the RNA was cleaned up before it left the nucleus, and now we're calling it mRNA, that messenger RNA. So we're going to move on from transcription into translation. So translation involves going from the messenger RNA to the amino acid sequence or the polypeptide to the protein, because that's what we're really trying to do here, is make proteins. And so this happens on the ribosomes, as we talked about in chapter 4. It involves another RNA molecule. And so we have a new kind of RNA. This time it's called transfer RNA or tRNA. And it's just an individual molecule. So we had polymers of DNA and polymers of RNA. Transfer RNA or tRNA is an individual molecule. This is an image of a transfer RNA molecule. And so you can see it has a few components, and we're going to just introduce ourselves to this tRNA here so that we can understand its role in translation a little better. So its job is to match an amino acid to the corresponding messenger RNA, so to the codon, if you will. Remember, this is what you were doing with that dictionary. So transfer RNA's structure allows it to convert from one language to the other. So down here we have nucleic acid language, and up here we have amino acid language. This is the amino acid that the transfer RNA is carrying, and down here we have the messenger RNA in pink. We have a codon, that 3-letter nucleotide base sequence as a codon, a triplet, if you will. Almost looked like it went to 4 there. Forget that one there. And then we have this other cool part here. We have an anticodon, which is part of the transfer RNA that allows the transfer RNA to bind to the messenger RNA. So here, this is the anticodon. It matches the codon with that specific base pairing. It says it's complementary in sequence. And so we have the A's and the U's pairing and the C's and the G's pairing. Again, because we're in RNA land, not DNA land, we don't have thymine, we have uracil. But we've been doing a lot of that base pairing on through all of these processes. In order to understand how translation happens, we also need to understand a little bit about the ribosomes. And so ribosomes are made of a third type of RNA. When you did your discussion of what the nucleolus contains, you said it was the recipe, or it was the information for making ribosomes. And that information is ribosomal RNA or rRNA. It makes ribosomes. And so we have 3 kinds. We have messenger RNA, we have transfer RNA, and now we have ribosomal RNA. Ribosomal RNA really just factors into making the ribosomes. We're not going to really talk about that process, but we have been talking about how messenger RNA relates to transfer RNA, and that's really the story of translation. So ribosomes are made of that third kind of RNA. If we were to take a look at a ribosome, there are 2 parts of a ribosome, a large subunit, which is larger, and a small subunit, which is smaller. The small subunits have a really important bit on them where they hold the messenger RNA. So they have a binding site for that messenger RNA. And so I'm going to switch to that color. The messenger RNA would sit between the small and the large binding site, or sorry, the large and the small subunit, being held by the small ribosomal subunit. Okay, the other thing on the large subunit is that it has 2 locations to hold the transfer molecules, the transfer RNA, the tRNA. So these binding sites for the tRNA are named the active site, or the A site, and the polypeptide, or the polymer side. So polymer. Or you could think of it as the peptide site. So the A site is where, the tRNA is going to actively come in. The tRNA, the transfer RNA molecule that's holding the protein, or you can think of it as protein, so many things that P could start with, is in that P site of the large subunit. So we have an introduction to transfer RNA. We have an introduction to our ribosomes with its small and large subunit and the active and the polymer protein or polypeptide site, the P site. And now we need to really understand how this all comes together. This is an image trying to illustrate how they all come together. And so, like I said, we have the small subunit. Switch back. So this is the small subunit of the ribosome. We have the large subunit of the ribosome. We have the messenger RNA being held by the small subunit. The messenger RNA has those codons on it. And now we're starting to understand the transfer RNA molecules. So the transfer RNA molecules had the anticodons, right? Those complementary base pairs, 3 of them. And it has the amino acid as part of it. So here is the active site, and here is the protein site or the polypeptide or the polymer holding site. Because as these come in, this is, you know, all of the stages or the parts of the play that ends up being translation. Okay. So in these ones, in stage 1 of translation, we have initiation. And initiation brings together what we need for protein synthesis to begin. And so hopefully we have enough of a primer of all the parts that this explanation can actually make sense to you now. So initiation is going to happen in 2 steps. The first step is all about the messenger RNA and the small subunit. And the first tRNA binding to the messenger RNA at the start codon. And so we have the first tRNA binding to the messenger RNA. So up here, all about the small subunit. The second stage of initiation is more about the large subunit. And so the large subunit now joins the small subunit with its messenger RNA, and the first tRNA is going to go into that P site, that protein site, the peptide chain, whatever P was standing for in your world, the first tRNA goes into the P site. It should be at the start codon. So if we remember, AUG coded for methionine and was the start codon, and the first transfer RNA has the anticodon UAC because that's what would pair with my start codon. Okay, so let's come back to this. Let's go back to that wacky slide where you were filling out the components of transcription. So if this is where we're at and we're filling in more information about the process of translation, here we have the messenger RNA codons. So the first codon, the start codon, is AUG. We recognize that when we were doing this with the codon dictionary. So the transfer RNA molecule would be holding the amino acid methionine. And if we take a look at the anticodon for this, it would be UAC because those would be the complementary bases to that codon. We could work through because the next one coming in, this is going to be the codon recognition from the next stage. Again, with these, we could come back to this with this idea that this is the process. So all of these amino acids had an anticodon that was bringing them in. And so, a transfer RNA that was bringing them in. And they all had an anticodon. So we could fill out the anticodon on this, the tRNA molecules that I'm drawing here. That anticodon of the transfer RNA that was holding leucine would be GAC. Same on down the line, we could come up with the transfer molecules that held all of these amino acids. And on down the way. So let's go back to our process of translation. We had stopped with the initiation phase. And so the initiation phase had the mRNA, the small subunit, the first tRNA with that anticodon of UAC that matched the start codon. And the transfer RNA would be holding that methionine. Okay, so now that we've got everything in place, that whole idea that we're going to move on, the active site is open at this point. It's available to hold the next tRNA. So we're done with initiation. The next stage, like we did in transcription, after initiation, we had elongation. And so the elongation phase, or the step of translation, really has three parts. So the first part of it is codon recognition. And that's where we're matching the anticodon on the tRNA to the codon on the messenger RNA. And again, you could flip back to that page where I drew those transfer molecules and their anticodon. So that codon recognition is what has to happen. So that means that the tRNA is going to come into the A site. And when we have two transfer molecules, two transfer RNAs beside each other, one in the P site and one in the A site, now we have the possibility of a peptide bond formation. That is the joining of the new amino acid to the chain. After we have the peptide bond formation, everybody moves one to the left, that translocation, they're going to move along. So the tRNA is released from the P site, and what was in the A site now moves into the P site. So it's kind of like a little machine that chugs along, moving the transfer RNA molecules out of the way when they've given up their amino acid to that growing polypeptide. And so that's what's happening. The polypeptide is going to keep growing until we end up with the ribosome reaching the stop codon. So from translocation, then we'd have more codon recognition, another peptide bond formed, the transfer molecules are going to move over one spot, and that's going to leave us open to the next codon recognition. Another peptide bond, another moving over, another codon recognition, another peptide bond, another moving over until we get to the stop codon. And so the stop codon is the end of elongation and the beginning of termination. So the third stage of translocation is termination, and termination is the completed polypeptide being released and the ribosomal subunits separating. There is a one-page overview at the end of the PowerPoint. There's also an image that is next. In class, I used to act this out, which is something unfortunately I can't figure out how to do over Zoom for us. So there is no in-class activity. I think in class I'm going to play the 3-minute video that has all the stars beside it on Moodle because it is such a good summary. It's probably the best one because it gives that idea of that animation of things. Before I play that video, or before maybe you break to play that video, here are a few other things that I put in to help you understand. This is an image that's not from our textbook, but it does have a little more movement to it. And so here you can see that idea of the small subunit, the large subunit in behind here with its A site and its P site. We have the messenger RNA being held by the small subunit, and the messenger RNA has codons on it. The transfer RNA have the anticodons attached to them and their amino acids. So here we have that idea of elongation. So we have these transfer molecules getting out of the way when they've given up their amino acid to that growing polypeptide chain. And here we have the new transfer molecules still holding their amino acids coming in for this codon recognition. This is trying to illustrate the peptide bonds being formed. And again, moving over from active site to the A site to the P site is that translocation. Again, one more way of representing it, the last slide. So here we have initiation, where we have the small subunit and the messenger RNA, and we have the first tRNA coming in with the start codon. The second stage of initiation, we had the large subunit, and the large subunit holding that first transfer RNA in the P site. Elongation, we'd have codon recognition, we'd have peptide bond, and then we'd have translocation. And that would be a process, like a cycle, keep going on and on until we ended up with a growing or fully formed polypeptide chain. Termination would be when after it hit the stop sequence and releasing of the polypeptide, the finished, and the ribosomal subunits coming back apart. So again, we'll watch the video and then carry on with the last part of this class. So now that we understand a little bit more about the function of DNA and how it actually codes for making these proteins, we're better set to understand how things can go wrong. And so a mutation we talked about way long ago when we were talking about the dangers of isotopes. A mutation can be a change in the genes. It changes the meaning of the genes. And so mutations could be a base substitution, so just a different nitrogenous base coming in, the wrong nucleotide, if you will. It could also be that we have extra information or missing information or extra information. And so in these ones, sometimes we can have a big effect on the protein that's going to be made, but sometimes we don't have a huge effect. So here, if we have the wrong base put in, the wrong nucleotide used, it would really depend if there was an amino acid change for that, because remember we had that redundant nature of our codon dictionary. The other ones, the deletion or the insertion, these ones can have a bigger effect because they're going to change the reading frame. They're going to change the grouping of the nucleotides so that they're actually coding for different amino acids potentially. So they could lead to significant changes in that amino acid sequence. It's everything downstream of that mutation. So we got a picture here that I think can help illustrate these ones. So again, this would be the simple wrong nucleotide coming in and the difference between normal hemoglobin and sickle cell hemoglobin. The other thing we could have is this stuff downstream. And so if we have a missing nucleotide there, everything downstream is going to be the wrong amino acid. And so this time, that would be a big change. That would maybe be a pretty serious mutation. So the sequence or the summary of this is that mutations could be of no consequence or of serious consequences. They can be spontaneous, just for random errors. I don't know if you made any mistakes in your slide where we were practicing that, but it's pretty easy to do for us. So I'm sure at a cellular level, it's also difficult. It could also be induced. And so this is where we talk about, that idea of isotopes and the danger of that, or chemicals in the environment. And so, mutations are something that were probably brought up in the Chapter 9 material, but now that we understand the process of how DNA becomes proteins, we have a deeper understanding of how information can be stored and expressed or altered and have potentially awful consequences.